Hey everyone, and welcome back for another deep dive. Today we are going to be looking at a topic that's really important in science, even though it's often overlooked.
Mm-hmm.
And that is sample preparation for chemical analysis.
Yeah.
You might be wondering why this is relevant to you.
Right.
Well, if you are prepping for a meeting, or maybe you're trying to get up to speed in a new field,
Mm-hmm.
or, maybe you're just curious about how scientists actually get reliable data from these messy real-world samples, then this deep dive is for you.
That's right.
Think of it like preparing ingredients for like a gourmet meal.
Yeah.
The quality of the final dish that you serve depends heavily on how you prepped those ingredients.
Absolutely.
But in our case, the dish is accurate scientific data.
That's a good analogy.
So to get us started, I am joined by an expert in analytical chemistry.
Hi, everyone.
So before we jump into the specifics, I'm
Yep.
curious, can you give us a sense of just like the overall whys?
So-
Like, why is sample preparation so important? What are we really trying to achieve here when we do this?
Well, the goal of sample prep is to take a complex, real-world sample
Okay.
and transform it into something that can be accurately analyzed.
Mm-hmm.
So imagine you're trying to count the different types of fish in a really murky pond.
Okay.
You need to clear the water first to get a good view right.
Right.
Sample prep does something similar.
Mm-hmm.
It removes anything that could interfere with the analysis so that we can focus on the specific components that we are interested in.
Gotcha. So it's about creating like a clean slate for the analysis and making sure that the data we get is accurate and reliable.
Exactly.
Okay. Starting to see why this is so crucial.
Absolutely. And there is like a whole toolbox of different techniques that are designed to tackle this challenge.
I bet.
Our sources mention everything from protein precipitation
Yeah.
to solid phase extraction,
Oh, wow.
derivatization, digestion,
That's a lot.
a bunch more.
Yeah. It sounds like there is a technique for every single type of sample and every single type of analysis.
Absolutely.
So where do we even begin?
Let's start with one of the most common techniques,
Okay.
protein precipitation.
Okay.
And this is often used when researchers need to isolate specific proteins from complex mixtures,
Mm-hmm.
like blood or tissue.
Okay, that makes sense. I mean, proteins are involved in so many different biological processes, so I can see why isolating them would be important.
Absolutely.
Yeah.
So the principle behind protein precipitation is to disrupt the protein's hydration shell,
Okay.
which is a layer of water molecules that surrounds and stabilizes the protein.
Gotcha.
By disrupting this shell, we reduce the protein's solubility,
Okay.
causing it to aggregate and precipitate out of the solution.
So we're like coaxing the protein out of hiding by changing its environment.
That's a good way to put it.
Yeah, okay, cool.
And there are a variety of ways to achieve this.
Mm-hmm.
Our sources mention three main types:
Okay.
acid precipitation, salting out, and alcohol precipitation.
Let's start with acid precipitation. I have to admit, the idea of using acid sounds kind of harsh, especially when we're dealing with these like delicate proteins.
Yeah, it can be.
Yeah.
So acid precipitation involves lowering the pH of the solution,
Okay.
which can change the charge of the protein, leading to precipitation.
Mm-hmm.
A commonly used acid is trichloroacetic acid, or TCA.
Okay.
It's very effective, but it can also denature the protein,
Oh, no.
meaning it might lose its biological function.
So it's not necessarily ideal if we need to keep the protein functional.
Right, so this method is best used when you need to quickly and efficiently remove proteins,
Mm-hmm.
perhaps to prepare a sample for analyzing something else.
Gotcha. So it's a powerful tool,
Yeah,
but not always the best choice if you need your protein to remain active.
exactly.
Okay, so what about salting out then?
So salting out is a gentler approach.
Okay.
By adding high concentrations of salt,
Mm-hmm.
we compete with the protein for water molecules.
Ah.
This disrupts the hydrogen bonds that keep the protein soluble, causing it to aggregate and precipitate.
So it's like gently nudging the protein out of the solution without damaging it.
Exactly. And the beauty of this method is that it often preserves the protein's function.
So that makes salting out the go-to method when you need to purify a protein but keep it intact.
Exactly.
Really interesting how each technique has its own strength and weaknesses depending on what you're trying to do.
Mm-hmm.
So what about alcohol precipitation? That sounds like it could be tricky.
Alcohol precipitation works by reducing the protein's hydration,
Okay.
similar to salting out,
Mm-hmm.
causing it to clump together.
Okay.
The challenge is that it can also precipitate other compounds, like salts and lipids,
Oh,
which might interfere with your analysis.
so it's a bit less targeted than the other methods.
Yeah.
And you might end up with a less pure sample.
That's right. It's important to choose the right tool for the job,
Mm-hmm.
considering both the protein that you're trying to isolate and the overall goal of your analysis.
For sure. So our source material actually throws in a couple of case studies here, almost like a pop quiz
Okay.
to test our understanding.
I like it.
So do you want to walk our listener through those?
Sure, let's put our listener to the test.
Okay.
So imagine you are tasked with purifying an active enzyme at room temperature.
Okay.
Which protein precipitation method would you choose and why?
That's a good one.
Yeah.
I'm thinking about the need to preserve the enzyme's activity, so I'm leaning towards...
Hold that thought.
Okay.
Let's give our listener a chance to ponder this.
All right.
Here's the second scenario. You need to measure mycotoxin levels in human blood using a technique called liquid chromatography, mass spectrometry, or LCMS.
Okay.
Which method would be most suitable for this case and why?
Okay, so those are two very different scenarios, each with their own challenges.
Right.
I'm curious to hear what our listener comes up with. But for now, let's move on to another fascinating technique.
Okay.
Solid phase extraction, or SPE.
SPE is incredibly versatile.
Or,
It allows us to separate analytes based on their chemical properties,
okay.
such as their polarity, charge, or even a combination of factors.
Okay, I'm intrigued. How does this work in practice?
Yeah.
Our source mentions using a pump, which makes me think of some kind of elaborate laboratory setup.
It's actually simpler than it sounds.
Okay.
The basic setup involves passing a sample through a column filled with a solid phase, which is a material that selectively retains certain compounds.
Okay.
By carefully choosing the right solid phase and solvents, we can wash away unwanted components while retaining our target analyte.
So it's like a microscopic filter that can recognize and trap specific molecules based on their chemical properties.
Exactly.
Okay.
Imagine a microscopic sorting machine, separating molecules based on their stickiness to the solid phase.
Love it.
Yeah.
This level of selectivity is really crucial for isolating our target analyte from a complex mixture.
Absolutely. And the pump that you mentioned simply helps to control the flow of liquids through the column, ensuring a consistent and efficient extraction process.
That's a great way to visualize it.
Yeah.
So SPE is all about manipulating those interactions between our analyte and the solid phase to achieve a separation. It sounds like a very powerful technique.
It is, and it's widely used in various fields, from environmental monitoring to pharmaceutical analysis.
Wow.
Our source material even mentions some specialized applications of SPE, like online SPE and microdialysis.
Oh, microdialysis. That one caught my eye. It sounds incredibly futuristic.
Yeah, microdialysis allows us to measure the concentration of analytes directly in living systems,
Wow.
also known as in vivo measurements. It's like having a tiny, real-time sensor implanted in a living organism.
Whoa, that's taken sample preparation to a whole new level.
It is.
Wow, imagine the possibilities.
It's truly groundbreaking.
Yeah.
Microdialysis opens up a whole new world of research, allowing us to study dynamic processes in real time without having to sacrifice the organism.
It sounds like science fiction come to life.
It does.
I'm eager to hear more about this.
We can definitely delve into more detail later.
Okay.
But for now, let's shift gears a bit and talk about another crucial sample preparation technique.
Okay.
Derivatization.
Derivatization. Okay, that one sounds a bit mysterious.
Yeah.
What exactly are we doing to the sample in this case?
Derivatization is all about chemically modifying the analyte,
Okay.
sort of like giving it a makeover
Okay.
to enhance its properties for analysis.
A makeover for molecules. I love it, but why would we want to do that?
Well, imagine you have an analyte that's not very volatile,
Okay.
meaning it doesn't easily turn into a gas. This can be a problem for techniques like gas chromatography, which rely on separating compounds in their gaseous state.
Okay, I see the challenge. So derivatization helps us overcome those limitations
Yes.
by making the analyte more amenable to the analysis technique?
By attaching specific chemical groups, we can increase volatility for gas chromatography,
Okay.
improve stability for long-term storage, or even enhance detectability by adding a flag
Oh.
that instruments can easily recognize.
So it's like giving the analyte a special outfit that makes it stand out in a crowd.
Exactly.
Okay. And you mentioned something about chiral separation earlier.
Yes.
How does derivatization play a role in that?
Ah, chirality.
Yeah.
It refers to the handedness of molecules. So some molecules exist in two forms that are mirror images of each other, but not superimposable,
Okay.
like your left and right hands.
Okay.
They look similar, but they can't be perfectly aligned.
So they're like molecular twins, but with a slight twist?
Precisely. And these chiral twins can have different biological effects.
Oh, wow.
So derivatization can help us separate and analyze these individual forms,
Okay.
which is crucial in fields like pharmaceuticals, where the two forms of a drug might have drastically different effects in the body.
Wow, that's incredible. It's amazing how such a subtle difference in molecular structure can have such significant consequences. It's a good reminder that chemistry is happening at a level we can't even see.
It really is.
So speaking of things that we can't see, let's shift gears to a technique that helps us break down complex samples into smaller, more manageable pieces.
Okay.
Digestion.
Digestion.
Our sources mention different types of digestion:
Uh-huh.
enzymatic, acid, and microwave.
Right.
So let's start with enzymatic digestion.
Sure.
What's happening at the molecular level here?
So enzymatic digestion uses enzymes, which are nature's catalysts,
Okay.
to break down large molecules like proteins or carbohydrates into smaller fragments.
Okay.
It's like having a tiny molecular chef in our sample, carefully chopping up those complex ingredients.
I love that analogy.
Yeah.
And I imagine different enzymes have different cutting preferences, just like a chef would use different knives for different tasks.
Exactly. We choose specific enzymes based on the target molecule and the desired outcome of the digestion. For example, if we want to analyze the amino acid sequence of a protein, we choose an enzyme that cleaves the protein at specific points along its chain.
So it's not just about breaking things down, it's about doing it in a controlled and precise way.
That's right.
Okay, what about acid digestion? That sounds a bit more intense.
Acid digestion uses strong acids like hydrochloric acid or nitric acid
Okay.
to break down complex matrices and release analytes into solution.
Okay.
It's a bit like using a powerful solvent to dissolve everything and free up the molecules we're interested in.
Okay, so it's a more forceful approach, but it gets the job done.
It does. Acid digestion is often used in elemental analysis, where we want to measure the concentration of specific elements in a sample.
Gotcha. What about microwave digestion? Is that like a super speedy version of acid digestion?
You could say that microwave digestion uses microwave energy to heat the sample and acid mixture, accelerating the digestion process.
Okay.
It's faster, more efficient, and often results in better recovery of the target analytes.
It's like using a pressure cooker for sample prep,
Exactly.
speeding things up and intensifying the reaction.
Yeah.
Now, before we move on, remember those case studies we presented earlier?
Yes.
Let's see if our listener has figured out the best protein precipitation method for each scenario.
All right.
So let's start with the first one. Purifying an active enzyme at room temperature. Any guesses out there?
So the key here is preserving the enzyme's activity.
Mm-hmm.
So the ideal method would be salting out.
Okay.
It's a gentler approach compared to acid precipitation, which could denature the enzyme.
That makes sense. Salting out allows us to gently nudge the enzyme out of solution without compromising its function.
Exactly.
What about the second scenario? Measuring mycotoxin levels in blood using LCMS.
So in that case, we need to consider that LCMS is a very sensitive technique.
Mm-hmm.
Even trace amounts of interfering compounds could skew the results.
So we need a method that removes as much noise as possible,
Right.
even if it means sacrificing some selectivity.
Exactly. Alcohol precipitation might be a good choice here. While it might not be as specific as other methods, it's effective at removing a broad range of potentially interfering compounds,
Okay.
ensuring accurate mycotoxin measurements.
So it's a trade-off between selectivity and thoroughness. And in this case, thoroughness wins.
Yeah.
It's fascinating how each technique has its own strengths and weaknesses, and choosing the right one depends on the specific requirements of the analysis.
Absolutely. And speaking of specific requirements, let's talk about a technique that was developed for a very specific purpose.
Okay.
Queachers.
Queachers. That sounds like a made-up word. What does it stand for?
It stands for quick, easy, cheap, effective, rugged, and safe.
It certainly is.
Quite a mouthful, right?
But it also sounds like the ideal sample preparation technique. What is the story behind this method?
So Queachers was specifically developed for analyzing pesticide residues in food.
Okay.
It's a relatively simple two-step process that involves extraction followed by cleanup.
So it's like a streamlined approach to getting rid of those unwanted pesticide contaminants.
Exactly the extraction step uses a combination of salts and organic solvents to pull the pesticides out of the food matrix.
Mm-hmm.
Then a cleanup step using a special sorbent removes any interfering compounds, leaving you with a clean extract for analysis.
So it sounds very efficient and targeted. Is Queachers only used for pesticide analysis, or can it be applied to other fields as well?
While it was initially developed for pesticides, Researchers are starting to explore its applications in other areas, like environmental monitoring and pharmaceutical analysis.
Wow.
It's a testament to its versatility and effectiveness.
It's great to see how techniques developed for one area can be adapted and applied to new challenges.
hmm
It speaks to the creativity and problem-solving nature of science.
It does. And speaking of adapting and innovating,
Yes.
let's circle back to filtration, a technique we touched upon earlier.
Right, filtration, it seems so straightforward.
Yeah.
But our sources emphasize that selecting the right filter membrane is crucial. It's not
Oh,
just about size, it's also about the material the filter is made from.
that's a great point. Different membrane materials have different chemical properties and compatibility with various solvents and analytes.
So it's not a one-size-fits-all situation. We need to consider the specific requirements of our sample and analysis.
Exactly. For example, nylon filters are solvent resistant
Okay.
and work well with both aqueous and organic solvents,
Mm-hmm.
but they're not ideal for protein analysis because proteins can sometimes bind to nylon.
Ah, that makes sense. So if you were studying proteins, you'd want to choose a different filter material.
Exactly. Something like PVDF, which has low protein binding, would be a better choice in that case.
PVDF, is that like a super smooth surface that proteins just slide right off?
You could think of it that way. The choice of filter material really depends on the specific application.
Mm-hmm.
Our source material goes into detail about a variety of other membrane materials like PTFE, PES, and cellulose, acetate, each with its pros and cons.
It's fascinating how much thought goes into something as seemingly simple as filtration.
Yeah,
It's a reminder that every step in the sample prep process matters.
it truly does. And speaking of important steps, let's revisit derivatization, the molecular makeover we were discussing earlier.
Okay.
I know you were curious about how changing the chemical structure of an analyte actually helps with the analysis.
Yes, I'm still a bit fuzzy on that. Can you walk me through it again?
Certainly. Let's say we have an analyte that's not very stable at high temperatures.
Okay.
This could be a problem for techniques like gas chromatography, where the sample is heated to separate the components.
Right, okay. I see the issue. We don't want our analyte to break down during the analysis.
Exactly. Derivatization can help us overcome this by attaching a specific chemical group to the analyte.
Mm-hmm.
We can enhance its thermal stability, making it more robust during the analysis.
So it's like giving the analyte a protective shield to help it withstand the heat.
That's a great analogy.
Okay.
And in addition to improving stability, derivatization can also be used to increase detectability.
Yeah.
Some analytes are difficult to detect directly using certain instruments.
Mm.
But by attaching a tag that the instrument is sensitive to, we can enhance its signal and make it easier to quantify.
So it's like giving the analyte a beacon that makes it more visible to the instrument.
Precisely. Derivatization is a powerful tool that allows us to manipulate the properties of analytes to make them more suitable for analysis.
It sounds like there's a derivatization strategy for almost any analytical challenge.
There probably is. And that brings us to the end of our discussion on derivatization.
Okay.
We've covered quite a lot of ground today, haven't we?
We have. It's amazing to think about all the work that goes on behind the scenes
Yeah.
to make sure that scientific data is accurate and reliable.
It's a hidden world that's crucial to the advancement of science.
Absolutely. But before we conclude, I think there are a few more fascinating techniques we should touch upon.
I agree. Yeah, there are a few more, and some of these techniques are truly ingenious. Like, have you ever heard of solid phase microextraction, or SPME?
SPME. That rings a bell, but I can't quite place it. How does it differ from the solid phase extraction that we discussed earlier?
So it's a variation of SPE,
Mm-hmm.
but instead of like a packed column,
Yeah.
SPME uses a small fiber that is coated with a sorbent material.
So it's a miniaturized version using this tiny little fiber to do all the heavy lifting.
Exactly. That tiny fiber can be immersed directly in the sample
Mm-hmm.
or exposed to the headspace above it,
Okay.
which is perfect for capturing volatile compounds.
Oh, I can see how that would be useful for like analyzing the aroma of coffee or the fragrance of flowers.
You got it.
Yeah.
SPME is known for its simplicity, sensitivity, and its ability to work with a wide variety of samples.
Sounds like a very handy tool for analytical chemists.
Yeah.
So what other techniques are out there that we haven't discussed yet?
Well, one that's becoming increasingly popular is supported liquid extraction, or SLE.
Supported liquid extraction. Okay, now that one is completely new to me.
Yeah.
What's the basic idea behind that?
Think of it as a bridge between traditional liquid extraction, or LLE, and solid phase extraction.
Oh, okay. I'm falling so far. Remind me, what's LLE again?
So in LLE, you shake your sample with a solvent that doesn't mix with it,
Uh-huh.
like oil and water,
Right.
to extract the analyte.
Right. I remember now. It's like a simple concept, but I imagine it can be pretty messy and time-consuming.
Exactly. SLE addresses those drawbacks
Okay.
by using a porous support material like diatomaceous earth
Mm-hmm.
to hold a small amount of that extraction solvent.
So instead of shaking the whole sample with a large volume of solvent,
Right.
we're using a smaller, more contained amount.
Precisely. This reduces solvent use,
Mm-hmm.
speeds up the extraction, and minimizes the formation of emulsions.
Emulsions. Aren't those mixtures that don't separate easily, like mayonnaise?
Yes, they can be a real headache in the lab.
I bet.
SLE helps to avoid that problem,
Mm-hmm.
resulting in a much cleaner extraction process.
So SLE takes the best aspects of LLE and SPE.
Yeah.
It combined efficiency with reduced solvent use.
Right.
It's like a hybrid sample prep technique.
That's a great way to put it.
Yeah.
It highlights how scientists are always looking for ways to improve and innovate, even in something as fundamental as sample preparation.
It's been such a fascinating journey exploring this often overlooked aspect of science.
I agree.
I feel like I have a much better understanding of just how critical sample preparation is to research.
Absolutely.
As technology continues to advance, I'm sure we'll see even more innovative and sophisticated sample prep techniques emerge.
Oh, for sure.
But even with all of the fancy new techniques, it seems like the fundamentals of sample preparation will always be important.
Absolutely. No matter how advanced the technology gets, the goal remains the same. To ensure that our samples are properly prepared and ready for analysis, because that's the foundation upon which reliable scientific knowledge is built.
Well said. It's been an absolute pleasure diving deep into this world with you today.
The pleasure was all mine.
And to our listeners, thanks for joining us on this knowledge-packed adventure. Remember, the next time you hear about a groundbreaking scientific discovery, take a moment to appreciate the meticulous work that went into preparing those samples. Because without it, none of those discoveries would be possible.
Absolutely.
Until next time, keep exploring, keep learning, and stay curious. Welcome back to the show.
It's great to be back.
It's amazing how much depth there is to this whole world of sample preparation.
I know, right?
It feels like we've only just scratched the surface.
Yeah, there's always more to learn.
We've covered a lot, but you're right.
Yeah.
There's always more to explore.
For sure. And some of these techniques are truly ingenious. Like, have you heard of solid phase microextraction?
Solid phase microextraction.
Or SPME?
SPME, that rings a bell, but I can't quite place it.
Okay.
How does it differ from the solid phase extraction that we talked about earlier?
It's like a variation of SPE.
Okay.
But instead of using a packed column, SPME uses a small fiber coated with the sorbent material.
So it's like a miniaturized version,
Exactly.
using this tiny fiber to do the heavy lifting.
Exactly. And that tiny fiber can be immersed directly in the sample
Okay.
or exposed to the headspace above it,
Gotcha.
which makes it perfect for capturing volatile compounds.
Oh, I see. So this would be useful for, like, analyzing the aroma of coffee or the fragrance of flowers.
You got it.
Okay, cool.
Yeah, SPME is known for its simplicity, sensitivity, and ability to work with like a wide variety of samples.
Sounds like a pretty handy tool for analytical chemists.
Absolutely.
So what other techniques are out there that we haven't talked about yet?
Well, one that is becoming increasingly popular is supported liquid extraction, or SLE.
Supported liquid extraction.
Yeah.
SLE. Okay, that one is completely new to me.
Okay.
What's the basic idea behind it?
Think of it like a bridge between traditional liquid extraction, or LLE,
Okay.
and solid phase extraction.
Okay, I'm following so far, but just to refresh my memory, what was LLE again?
So in LLE, you shake your sample with a solvent that doesn't mix with it, like oil and water.
Right. And this extracts the analyte.
Right. I remember. Now, so it's a simple concept.
Yeah.
But I imagine it can get pretty messy and time-consuming.
Exactly. And SLE addresses these drawbacks
Okay.
by using a porous support material like diatomaceous earth
Mm-hmm.
to hold a small amount of that extraction solvent.
So instead of shaking the whole sample with a large volume of solvent, we're just using a smaller, more contained amount.
Precisely. And this reduces solvent use,
Okay.
speeds up the extraction, and minimizes the formation of emulsions.
Emulsions.
Yeah.
Aren't those the mixtures that don't separate easily, like mayonnaise?
Exactly. They can be a real headache in the lab.
Oh, I bet.
SLE helps to avoid that problem,
Mhm.
resulting in a cleaner extraction process.
So SLE kind of takes the best aspects of both LLE and SPE.
Yeah,
It combines efficiency with reduced solvent use.
right.
It's like a hybrid sample prep technique.
That's a great way to put it.
Yeah. And it highlights how scientists are always looking for ways to improve and innovate, even in something as fundamental as sample preparation.
It really does. It's been such a fascinating journey exploring this often overlooked aspect of science.
Yeah, I agree.
I feel like I have a much better understanding of just how crucial sample preparation is to research.
Absolutely.
And as technology continues to advance,
Hmm.
I'm sure we'll see even more innovative and sophisticated sample prep techniques emerge.
Oh, yeah, for sure.
But, even with all these fancy new techniques, it seems like the fundamentals of sample preparation will always be important.
Yeah, I think so too. No matter how advanced the technology gets, the goal remains the same,
Right.
which is to ensure that our samples are properly prepared and ready for analysis, because
Mhm.
that's really the foundation upon which all reliable scientific knowledge is built.
Well said. It's been an absolute pleasure diving deep into this world with you today.
Likewise.
To our listeners, thank you so much for joining us on this knowledge-packed adventure.
Thanks, everyone.
And remember, next time you hear about some groundbreaking scientific discovery, take a moment to appreciate all of the meticulous work that went into preparing those samples.
Absolutely.
Because without it, none of those discoveries would be possible. Until next time, keep exploring, keep learning, and stay curious.